It is my great pleasure to welcome here our international mentor, Dr. Norbert Pardi from the University of Pennsylvania, and Monica Kirici will introduce our lecturer. So please, Monica, go on.
Thank you very much. It's also my pleasure. Norbert Pardi holds a PhD in biochemistry and genetics from the University of Szeged, Hungary. He has been working at the University of Pennsylvania since 2011 and currently holds an associate professor position at the Department of Microbiology of the Perelman School of Medicine. He received training from Dr. Karikó Katalin, Dr. Drew Weissman at the University of Pennsylvania before he started his own laboratory. His research interest is the development of mRNA-based therapeutics with a particular focus on new generation infectious disease vaccines. He explored the development of novel vaccine platform using nucleoside-modified mRNA in lipid nanoparticles and used it to generate highly effective vaccines targeting various pathogens, influenza virus, coronaviruses, malaria, and others. Dr. Pardi is a pioneer of the nucleoside-modified mRNA vaccine technology and published milestone papers in the field. Hogyha lehetséges, akkor magyarul folytatnám, ugyanis nekem van egy kicsit személyesebb laudációm is Norbiról. Nagyon sok ponton kötődünk egymáshoz. Pardi Norbert 1981-ben született Szolnokon, de Kisújszáson töltötte a gyerekkorát, ahonnan Karikó Katalin is származik, a saját férjem is. Így mind a ketten ismerjük azt a közeget, és nagyon szeretjük ezt a kisvárost, és Norbi is azt mondta, hogy ott szeret élni igazán. Gyermekkoráról így vallott, gyerekként a természet szeretete terelt a biológia irányába, az erdők és rétek ott voltak a házunk mellett, ott nőttem fel, rengeteget jártam a természetet, horgáztam, nagyon gyorsan megismertem az állat- és növényvilágot, nagyon szerettem David Attenborough könyveit és műsorait, és így már akkor eldöntöttem, hogy a természettudományokkal szeretnék foglalkozni. Kisújszálláson a Kossuth Általános Iskolába járt, majd a szolnoki Széchenyi István gimnáziumban biológia angol tagozaton tanult. Az érettségi után a Szegedi Tudományegyetemre nyert felvételt biológus szakra, molekuláris biológia genetika szakirányon folytatta a tanulmányait. Így vallott erről. Már az egyetemi évek alatt éreztem, hogy szakmai tanácsokra van szükségem, ezért megkerestem Karikó Katalint, hiszen ő is kisújszállási, rendkívül nyitott volt, amikor hazalátogatott, mindig szakított időt arra, hogy tudományról beszélgessünk, és megosztotta velem a legfrissebb kutatásait, néha még a publikáció előtt álló eredményeket is. Ez nagyon inspiráló volt számomra. 2004 és 2011 között PhD hallgatóként folytatta kutatómunkáját Boros Imre professzor csoportjában, biokémiai, molekuláris biológiai, genetikai kísérleteket végzett, és mi ekkor ismerkedtünk meg igazából Pardi Norberttel, mindketten Boros professzor úr vezette szakmai műhelynek voltunk a tagjai. Pardi Norbert most professzor, akkor PhD hallgató volt, én pedig friss postdoc voltam. Nagyon sokat beszélgettünk, nem csak szakmai dolgokról, de más fiatal kutatók életében felmerülő dilemmákról, mi a legjobb út egy fiatal kutató előremenetele szempontjából. Mi vezet a sikerhez? Annak idején nem voltak ilyen szervezett fiatal kutatói képzések, mint ez a Tudós Akadémia. Mindenki magától próbálta a kutatói léthez kapcsolódó legfontosabb dolgokat elsajátítani, mint megfelelő angol nyelvtudás, külföldi tanulmányutak vagy hazai nemzetközi kapcsolatok. PhD értekezését 2011. április 11-én védte meg, én ott voltam. Disszertációjának a címe a Drosophila melanogaster P53 szumoilációjának molekuláris biológiai és genetikai vizsgálata. Hogyha esetleg nem tudnátok, akkor a szumoiláció az egy poszttranszlációs fehérje módosítás, nagyon hasonlít az ubiquitilációhoz, de nem a fehérjék lebontásának a feladata. Pardi Norbert 2011-ben csatlakozott Karikó Katalin és Drew Weissman laboratóriumához a Pennsylvania Egyetemen. A laboratórium akkoriban nagyon kicsi volt, ami nagyon előnyösnek bizonyult, 5-6 ember dolgozott együtt, és ez lehetőséget adott a nagyon szoros szakmai párbeszédre. Karikó és Weissman kezei alatt dolgozni hatalmas megtiszteltetés és egyben fantasztikus lehetőség volt, ezt mondta róla annak idején Norbert. Posztdoktori időszaka nagyon jól sikerült, nagyon sok cikket publikáltak Karikó Katalinnal és Weissmannal, a módosított nukleozidokat tartalmazó hosszú RNS-ek in vitro transzkripciójáról, ennek a kinetikájáról, illetve arról, hogy hogyan lehet ezeket a molekulákat lipid nanopartikulumokba csomagolva a szervezetbe juttatni, és így vakcinálásra felhasználni. A többi pedig azt hiszem, hogy történelem. Pardi professzor ma saját csoportját vezeti, a Pennsylvania Egyetem orvosi karán, ennek meghatározó kutatója, nem csak mRNA-s alapú vakcinák fejlesztésében volt forradalmi áttörése a kutatómunkájának, hanem új irányokat is mutattak az immunterápiák és vírusellenes stratégiák területén. Azt mondta erről az időszakról Norbert, nagyon büszke vagyok a csapatomra, amely nemzetközi, rendkívül tehetséges emberekből áll. Külön öröm számomra, hogy több magyar kollégám is van, akik Szegedről érkeztek, fantasztikus munkát végeztek. Ezzel is szeretném bizonyítani, hogy a magyar tehetségek világszerte megállják a helyüket. Rendszeresen publikál a professzor úr nagy presztízsű tudományos lapokba, Nature, Science, PNAS, ha jól számoltam, akkor fiatalkora ellenére 18 Nature cikke van, több mint 16 technológiai újítást talált fel, melyek szabadalomhoz vezettek, köztük a Zika vírus elleni immunválasz kiváltására szolgáló nukleozidmódosított RNS felhasználására, és 2021-ben ennek következtében a Gábor Dénes feltalálói díjat is megkapta, ugyanebben az évben a rangos Bayel díjat is megkapta. Már posztdoktori időszaka alatt is fiatal kutatói díjakat nyert el. Szerintem díjait, eredményeit, cikkeit hosszasan lehetne sorolni. Egy biztos, hogy Pardi Norbert neve az mRNA-s alapú vakcinák fejlesztésével és a jövő orvosi áttöréseivel fort össze, és például bizonyítja azt, hogy kitartással, elkötelezettséggel messzire lehet eljutni, és hogy a tudományvilága izgalmas, inspiráló lehetőséget kínál azoknak, akik hajlandóak keményen dolgozni. Szeretném megköszönni, hogy elvállaltad nemzetközi mentorságot ebben a Tudós Akadémiában, és ezért szeretném átnyújtani neked ezt az oklevelet, és ezt a kis ajándékot.
Nagyon szépen köszönöm, angolul folytatni, de nagyon szépen köszönöm a több mint kettes és részletes bemutatást, és az utolsó mondatoddal, azzal pedig külön nagyon-nagyon egyetértek, hogy tényleg úgy gondolom, hogy kemény munkával, odafigyeléssel, meg azzal, hogy az ember együtt tud dolgozni másokkal, és nem csak a nehézségeket osztjuk meg, hanem a sikerekben is mindig megosztjuk, vagy mindig elismerjük azt, hogy nem egyedül csináltunk valamit. Ha tényleg nem egyedül csináltunk, az úgy gondolom, hogy egy jó tanács a jövőre nézve, szóval hogy próbáljunk minél nagyvonalúbbak lenni az együttműködő partnerekkel. Jó, akkor átváltok angol nyelvre. Az előadásom az kb. Én azt gondolom, hogy 40 perc körül van, tehát mindenképpen lesz időnk kérdésekre, amit akár magyarul, akár angolul fel lehet tenni. Okay, so thank you very much for coming and listening. Today I'm going to tell you about the history of messenger RNA, particularly nucleoside-modified messenger RNA-based vaccines and therapeutics in general. I will show in the first part and longer part of my talk, I will tell you about the most important experiments, breakthrough studies that really led to the birth of this new platform technology, proved to be very, very efficient during the COVID-19 pandemic and really saved millions of lives. And after the historical parts, historical overview, I will tell you a little bit about the current standing of the field. I think this is something that you might be interested in. And then I will also tell you about the hurdles that need to be overcome to open new avenues for this platform technology. So let me start with showing a cartoon about the central dogma of molecular biology. The central dogma of molecular biology tells you about the flow of genetic information. Genetic information is passed from DNA to RNA and then RNA from protein. This is not a perfect cartoon because it doesn't show that it can go the other way around. So you cannot make DNA from protein, for instance. The reason why I'm showing this to you is that just want to mention that all three platforms, DNA, RNA, and protein, can be used for therapy, and messenger RNA is the newcomer in the field. It was discovered in 1961, and then it took 60 years, 6 decades to get approval for the first messenger RNA-based drug. I don't know if this 60 years is a long time or if it's not a long time. Probably it's very subjective. But what I can tell you about the reasons why it took 6 decades to get approval for the first mRNA-based drug. Very subjectively, I won't be able to tell you about everything because I don't have the time, but I will just tell you a bunch of reasons. So again, very subjectively, I believe that we can split these 60 years into 30 years and then roughly 30 years and then another 30 years. In my opinion, the first 30 years was between 61 and 1889. And it's very important to understand that even if you discover something, it really takes time until you understand how it exactly works. So again, messenger RNA was discovered in 1961, but people didn't know anything about it. They just discovered the molecule, but they didn't know anything about the structure. And obviously, if you want to use something for therapy, you have to understand, you have to know how it looks like, and then you can think about the different ways of manipulation, how to tailor it and make it into a medicine. So let me show you just two very, very important discoveries or invention that really help to learn more about RNA. Again, the most important thing that people could not make messenger RNA for a very, very long time. So it was very important that Doug Melton and Paul Krieg published a beautiful paper in Nucleic Acids Research in 1984. So this was 23 years after the discovery of RNA. And this was the very first study that showed that we can make IVT, in vitro transcribed, man-made messenger RNA. Until this point, people could isolate RNA from cells, but it was not possible to make it in the lab in an Eppendorf tube, let's say. So again, what they did was very simple. They made a plasmid DNA template. As you probably know, T7 RNA, sp6 or T7 RNA polymerase need a DNA template to be able to synthesize messenger RNA. So they made a plasmid DNA template that encoded the gene of interest. In this case, it was a human beta interferon gene. And of course, the plasmid contained an sp6 promoter for the sp6 RNA polymerase. And they just put together a very simple reaction, in vitro transcription reaction. They added the DNA template, the RNA polymerase, the 4 different nucleoside triphosphates, the building blocks of RNA, and of course, a capping enzyme. And they made a fully functional, beautiful RNA that encoded the human beta interferon. And then, to demonstrate that this RNA is functional, they microinjected this RNA into frog oocytes. I'd like to mention or to point out here that this was not a transfection, right? So they used the microinjector because at this time there was no mRNA transfection reagent available. So this is also very interesting because now we can use many different types and they all work well, but again 40 years ago this was the only way to deliver any nucleic acid into cells. So they injected the oocytes and then they demonstrated that human beta interferon was produced and they could measure it in the supernatant after 10-hour incubation, and again this just demonstrated that this was a fully functional RNA. So again, 23 years after the discovery of RNA, people could make messenger RNA, the product that they really wanted to make in the way they wanted to make in an Eppendorf tube. I told you about the need of delivery. Delivery is always a problem for any medicine. And it was 28 years after the discovery of messenger RNA when people could transfect mRNA into cells. So in this case, Phil Felgner's lab published a very nice paper in PNAS, and they demonstrated that when they use lipofectamine, they could transfect mammalian cells with a messenger RNA that worked really well. So again, this was almost 30 years after the discovery of RNA when people could transfect cells. This was only in vitro. So again, it was not a mouse study. The first mouse study that very clearly demonstrated that mRNA can be used in animals successfully. It was published in 1990, again by Philip Felgner's group, and they injected unformulated RNA encoding reporter constructs into mice intramuscularly, and they demonstrated protein production. So this was the first demonstration, first published demonstration of the functionality of a man-made messenger RNA. But despite the critical and very interesting findings of this beautiful science paper, most people remain skeptical about the therapeutic potential of RNA, and the field really focused on DNA-based and other approaches. What were the major concerns about RNA as a therapeutic platform? So first of all, the unstable nature of RNA. So compared to the other nucleic acid DNA, RNA is not that stable, and many people believe that it is really not possible to use it for therapy. I believe the most critical problem was its really high innate inflammatory capacity. So the fundamental problem is that your immune system cannot really distinguish between a therapeutic RNA and, let's say, a viral RNA, unless you modify the RNA that you want to use for therapy. So we have many different RNA sensors in different cellular compartments that recognize RNA. So if you just use an unmodified RNA, it will induce a very, very serious inflammatory response, because your body will believe that it is probably a viral RNA, it's a danger signal, and it induces very strong inflammation, cytokine storm, and everything else. So it was a critical, it was very important to figure out how to make the RNA less recognizable for the immune system. Allow in vivo translatability, so obviously you want to produce as much protein from the RNA as possible. If you want to do this, you need to optimize every single part of the RNA. You need to optimize the capping strategy, you need to optimize the untranslated regions, you need to optimize the coding sequence, the poly-A tail, everything. And of course, the last issue was insufficient in vivo delivery. Again, delivery is always a problem for any, or almost always a problem for any medicine. This was a big challenge also for messenger RNA-based therapies. Again, people could transfect RNA into cells, as I showed you from 1989, when lipofectamine, when people started to use lipofectamine and other reagents, but you cannot really use lipofectamine for sufficient and safe in vivo delivery. This is important. So the first three issues were more or less resolved by the early pioneers of the mRNA technology at the University of Pennsylvania, and you all know that Drew Weissman and Katalin Karikó received the Nobel Prize in medicine, physiology or medicine in 2023, almost 2 years ago. Let me tell you about the most important studies that the most important discoveries that really led to the Nobel Prize for them in this field. I will tell you about 3 papers very briefly. Most probably this experiment is the most critical, and it's really a beautiful study that was published in a very nice immunity paper 20 years ago. So what they did here is that they isolated RNA from different types of cells and different compartments. As you can appreciate, they isolated RNA from mammalian cells, but also from prokaryotic cells, and different types, nuclear RNA, cytoplasmic RNA, total RNA, and everything else. And then they transfected monocyte-derived dendritic cells, a cell type that is equipped with many different RNA sensors, and then they measured the inflammatory capacity of the RNA by measuring TNF-alpha production of these cells in the cell culture supernatant. Most importantly, look at the purple bar, they also used an in vitro transcribed RNA that they made in the lab. So the take home is basically the title of this slide. Natural RNAs are not equally potent activators of the immune system. So as you can see, some RNAs, such as transfer RNA from mammalian cells, did not induce any TNF-alpha production, but others, not surprisingly, the E. coli RNA, total RNA induced a very high level of inflammation, TNF-alpha production, and most importantly, as I told you, look at the purple bar, the unmodified RNA also induced a very high level of inflammation in dendritic cells. And the big question was why? So why do we see that some RNAs, such as transfer RNA, are not very inflammatory or not inflammatory at all, and others, such as an unmodified in vitro transcribed RNA or a mitochondrial RNA from mammalian cells, induce a very high level of inflammation. So what Katalin figured out, and this was Katalin, this was not Drew Weissman. So basically, she figured out that most probably the biggest difference between different RNAs is the natural modifications of the RNA. So there are more than 130 different types of modifications, such as base methylation, pseudouridylation, and others. I just listed some of them, and most importantly, pseudouridylation it's the most abundant modification, and you can find it in a bunch of different RNAs, such as in ribosomal RNA and transfer RNA. So this is why Katalin believed that this is primarily responsible for the different inflammatory capacity of different RNAs. And then again, she teamed up with Drew Weissman, a really fantastic mentor and fantastic immunologist, and again, together they were an excellent team, and they tested this hypothesis. Before I'm showing you the actual data, let me show you this figure about supporting the hypothesis. So if you look at it, on the x-axis you can see that it really shows the percentage of nucleoside modification in different RNAs, and you can appreciate that transfer RNA is highly modified, so almost 25% of the nucleosides in transfer RNA are modified. And then on the y-axis you can see the inflammatory capacity, so you can appreciate that the more, this is a reverse correlation, the more modified nucleosides you have in an RNA, the higher the inflammatory capacity. Again, transfer RNA, lots of modification, minimal, if any, inflammatory capacity. In vitro transfer, unmodified RNA, or E. coli RNA, or mitochondrial RNA, no or minimal modification, very high inflammatory capacity. So this was a clear clue. But they directly tested it, and it really turned out that when Drew and Katalin transfected monocyte-derived dendritic cells with an unmodified RNA, it really induced a high level of inflammation, but when they used different types of modifications, such as 5-methylcitidine modified or pseudouridine modified, pseudouridine, or actually a modified version of pseudouridine is used in the current generation of COVID-19 mRNA vaccines, you can appreciate that it did not induce or induce very low level of TNF-alpha production compared to the unmodified, and also lower level of IL-12 production. So the take-home from this experiment was that modified nucleosides can decrease immune activation, and this is very important for therapeutic applications. A couple of years later, they published a second paper that clearly showed that if you introduce certain types of modifications, again, such as pseudouridine, you can increase protein production from the RNA. So they transfected cell lines, 293 cells, or primary cells, murine dendritic cells, with unmodified RNA or different types of modified RNAs, and then they measured light, because this was a luciferase, firefly luciferase encoding RNA, and light production reflects the translational capacity. And again, it turned out that if you have pseudo-modified RNA, you can induce, I don't know, 8 to 9-fold higher level of protein production compared to the unmodified RNA shown with the blue bar. And you see the exact same thing in primary cells. So again, if you introduce these modifications, you can increase translation, which is really important, and you can decrease innate immune activation. That is a critical safety feature of the modified RNA. And this is really clearly necessary if you want to use it as a therapy or even as a vaccine. If you go back to this figure, I just want to show you one more time that, again, if you use modified RNA, you can decrease innate immune activation, but in many cases it's not zero. So particularly for IL-12, you see that there is still some immune activation induced by the modified RNA, and they really want to make Katalin, because Katalin wanted to use the RNA for therapy. She didn't really want to use it for vaccines. That was Drew Weissman. But Katalin wanted to use it for protein replacement, and obviously, and I will tell you more about it a little later, if you want to use it for protein replacement, you don't want any inflammation if possible. So they asked the question, what causes this residual immunogenicity in modified RNA? So what they were thinking was that most probably there are some contaminations in the in vitro transcribed RNA preparation. When you make an IVT mRNA, in vitro transcribed messenger RNA, the T7 or sp6 RNA polymerase always generates short abortive products. These are short single-stranded RNAs that can, since they are complementary, they can form double-stranded RNAs. And short double-stranded RNA or even long double-stranded RNA is very, very inflammatory. So it induces tons of inflammation. And it's very well known that we have many different double-stranded RNA sensors in different cellular compartments, such as toll-like receptor 3 in the endosome, but also we have more than 10 double-stranded RNA sensors in the cytoplasm. So Drew and Katalin believe that most probably this is responsible for the residual immunogenicity. And to directly test this, they did a Western blot experiment in which they used a double-stranded RNA-specific antibody called J2. And this J2 antibody recognizes double-stranded RNA in a sequence-independent manner. And what you can see here that when they apply this antibody, the RNA can be unmodified or modified. This preparation always contains double-stranded RNA contamination. And this is responsible for the residual immunogenicity that I showed you. So they developed an HPLC purification procedure to get rid of this double-stranded RNA and other contaminations. And this beautiful northern blot was done by Katalin Karikó with her own hands at the age of 50, 52, something like that. So it's pretty rare that professors do experiments at this age, but she did it, she made it. And you can see that indeed, if you use, and this is very important, if you use unmodified RNA, even if you HPLC purify it, it will induce inflammation. So you can see the interferon alpha, interferon beta, and the TNF alpha induction by unmodified RNA. Even if you purify, it's always inflammatory because toll-like receptor 7 and 8 recognizes uridines very, very well. And this is why modification is critical. But if you use pseudo-uridine modified RNA, if it's purified, you can get rid of this immune activation. So nothing is really measurable by northern blot. So by 2011 or so, they could make really high quality messenger RNA. And as you can appreciate, I don't know if you can see it or not, but hopefully you can, modified RNA transfection works much better than plasmid DNA transfection. So this was a very simple comparative study. 293 cells were transfected either with an pseudo-uridine modified RNA or plasmid DNA encoding EGFP, enhanced green fluorescent protein, and then the green cells were visualized. And you can see again that the transfection efficiency is higher when you use modified RNA. It can be close to 100%. It really depends on the cell type. And cells also produce much more protein. So this is one of the reasons why I do believe that RNA, it's not only a translational tool, but it's also a very, very important basic science discovery tool, because you can use it either for cell transfections. You transfect in the morning, you collect the cells in the afternoon, and again, you have very nice transfection efficiency in most cases. Of course, there are hard to transfect cells, so there are some exceptions, but to my best knowledge, almost any cell type can take RNA up. Again, in some cases it's lower efficiency, but in general it's pretty high. And you can collect your cells a couple of hours post-transfection, because the RNA is taken up directly to the cytosol, so the cells produce protein, and it's measurable as early as 20 minutes post-transfection. So you can use it for anything, basically, even if it's not a vaccination study, or even if it's not a mouse study. So this was the time, 2011, when I joined Drew and Katalin, and at this time the biggest, or the only main issue was delivery into animals, because again, Katalin wanted to use it for protein replacement. Drew wanted me to develop vaccines, but of course we need to vaccinate animals. We cannot do vaccination studies in cells, and so we had to figure out how to solve the delivery problem. And in the first 3 years of my postdoctoral period was really tough. As Moni mentioned, it was kind of fun in the sense that I could directly work with Drew Weissman. He was pipetting on my right, Katalin was pipetting on my left, I was in the middle. So we talked a lot about science, and again, I really learned from the best mentors. So it was fantastic. The lab was very small, so we really had a lot of time to talk about science and do experiments together, because at that time there was no money in the lab. So again, both Kati and Drew worked very, very hard in the lab. They were not just sitting in the office and do management work. It was a lot of wet lab research as well for both, and they were really, really good wet lab research scientists as well. So the first 3 years was really tough for me, because as a postdoc I really wanted to learn a lot, and this was successful, but I also wanted to be productive, and productivity at least partially means that you publish papers. And again, the first 3 years I really tried every single possible available reagents to deliver RNA, and many different ways to deliver RNA into animals, but none of them were successful. So I had enough data to publish 5 papers in the Journal of Negative Results, but we couldn't really publish anything in other journals until we bumped into a company called Acuitas Therapeutics, and this company is now very famous because their product is used in the Pfizer BioNTech COVID-19 vaccine. So we bumped into a publication from this company, and it turned out that they have very special RNA transfection reagents. They call them lipid nanoparticles, and they were developed to transfect or to deliver small interfering siRNA into animals. The reason why normal standard liposomes don't really work well in animals, because they are permanently positively charged. And this is a problem, because if you deliver a positively charged molecule into animals or humans, a lot of serum proteins will bind to the negatively charged serum proteins will bind to the positively charged lipids, so they can form clogs, and this will lead to a lot of adverse events. This is a very, very big problem. And the magic with these LNPs are that they are not positively charged, or they are almost uncharged under close to neutral pH. So for example, in the bloodstream, pH 7.4, so they are safe. So this was very important, but again, we didn't know if we can deliver messenger RNA as well. So after begging the company for several months, they formulated some firefly luciferase mRNA for me, and then I could inject mice in many different ways. systemic delivery, intravenous, intraperitoneal, or local delivery, intramuscular, subcutaneous, intradermal, even intratracheal. And then with this firefly luciferase encoding RNA, I could perform an in vivo imaging experiment in mice, and then it turned out that this mRNA LNP works really well in animals. So you can see the light really shows the translation from the RNA, and there are two different kinetics. When you deliver, it really depends on the LNP composition as well, but this is generally true, not always, but generally true. When you deliver it locally into the dermis, or even subcutaneously, or into the muscle, the vast majority usually stays there, but again, it's not always true in most cases. And you see protein production from the RNA for a longer period of time. Depending on the dose, it can be 10 days, 2 weeks, 3 weeks. And the other kinetics is after systemic delivery. Most LNPs target the liver after systemic delivery. It's excellent if you want to cure a liver disease, but it's really not great for everything else. And you usually see a very high level of protein production, but only for a short time. So let's say 2 days. Again, it really depends on the dose and the type of LNP, but this is generally true. And again, the take home is that the platform worked really well. And roughly 10 years ago, the nucleoside modified and purified mRNA lipid nanoparticle platform was born, and we and others started to use it for many different purposes. And most importantly, this platform technology is the basis of the very successful Pfizer, BioNTech, and Moderna COVID-19 vaccines. It's pretty much the same. There are obviously some differences in production. They don't reveal everything, all the details, but it's kind of similar. So let me tell you very briefly how we make this platform. So the RNA is encoded on a plasmid DNA template, so that's what you can see here. And every single part of the RNA, the untranslated 5' untranslated regions, the 3' untranslated regions, the poly-A tail is optimized. Everything is optimized. Once you perform the in vitro transcription reaction, you can enzymatically add a cap1 structure to the 5' end of the RNA. And as I told you, we purify the RNA to get rid of double-stranded RNA and other contaminations that would be recognized as pathogen-associated molecular patterns by the immune system, and they would induce a very high level of inflammation. So we don't have this. And as I told you, we replace uridines with N1-methylpseudouridine in most preparations that we use, particularly for vaccination, but also for other applications. We almost always use this one. And once this modified and purified RNA is made, it gets formulated into lipid nanoparticles. And this is, again, this is the platform technology, and you can use it for many different purposes, and there are pretty much endless possibilities for modifications and tailoring based on the application. I will tell you a little bit more about it. But again, 10 years ago, I was primarily interested in using this for vaccination, and then it turned out that it worked really well. We published the very first paper demonstrated that the modified, purified mRNA LNP vaccine technology works really well as a vaccine platform. So we published a paper about a Zika virus vaccine in 2017. And then after that, obviously now hundreds of papers demonstrated that it worked really well. And most importantly, what was important, because this was a mouse study and a non-human primate, a monkey study, but then it turned out that during the pandemic that it worked extremely well also in humans, and it's safe. And now there are many different clinical trials. Where are we now? I just took a screenshot from the webpage of Moderna showing you really just a tiny fraction of the vaccine clinical trials that are ongoing and Moderna. And now you can see that there are many. So many different versions of the COVID-19 vaccine, Zika vaccine, flu, RSV, and many others. And again, the list is longer. I just want to show you that now the field is flourishing. We'll see how it goes in the light of recent happenings with the Trump administration. But I'm still hopeful that there will be no gigantic harm. But Moderna has more than 50 ongoing clinical trials, as far as I know, just in the field of infectious disease vaccines. And so this is fantastic, and I'm pretty confident that we will see more mRNA-based vaccines that will be approved for human use in the near future. So infectious disease vaccines, the breakthrough happened. How about cancer? So during the COVID-19 pandemic, we heard from many sources that okay, the next breakthrough will be cancer vaccines using RNA, but obviously it doesn't happen so quickly because it's a very different scientific problem. For most prophylactic vaccines, particularly viral vaccines, you want to focus on antibody production, but this is not necessarily true for cancer vaccines. For cancer vaccines, you really want to induce a very strong CD8 T cell response. In most cases, for solid tumors, antibodies can also be useful. But in most cases you want to focus on CD8 T cells, and the requirement of CD8 T cell induction and antibody induction are different. So I think BioNTech is the leader in the field. They have many different candidates for different types of cancer, but again, no approved drug exists at the moment for humans. Hopefully, we will see some very promising data in the coming months and years. So this is about vaccines, but what else beyond vaccines? Therapeutics. So it's very important that the focus is certainly on vaccines, but you need to keep in mind that any protein can be encoded on messenger RNA that generates almost endless opportunities. But of course, it's very important to note that, and I'll tell you about it, that you cannot use the exact same platform for everything. So I'll just show you two examples, two publications. So we demonstrate, we and others. Now there are many, like 30, 40 follow-up papers on this showing that you can use messenger RNA therapy for monoclonal antibody therapy. You can encode, since antibodies are proteins, you can encode them on messenger RNAs. You can do it in several different versions. You can encode the light chain and the heavy chain and then formulate together and inject into mice or into animals or humans. But you can also make other formats, other antibody formats, if you will. People also started to use it for gene therapy. So you can encode genome editing constructs such as base editors on messenger RNA, and then you can fix certain types of diseases. There are probably hundreds of publications that really show that this is also doable. I just wanted to show you these two examples and showing you a screenshot again from Moderna's webpage that shows that they have many different candidates in different clinical phases. Some of them are in late phase 2, and there are many products. Again, this is just a fraction of the clinical trials, and there are many different types of diseases, genetic diseases, and other types of diseases that they really want to fix with messenger RNA-based therapy. You can check out the website and see what else they are doing. And let me tell you very briefly about the hurdles, because obviously for mRNA therapeutics we have issues. So I think I already told you about it in one of my talks in Szeged, that the lipid nanoparticle component of mRNA vaccines, it's critical not only for protecting RNA from RNases, but also and not only for delivery, but also for the addition of adjuvants an adjuvant component to the vaccine. So the lipid nanoparticle is highly inflammatory. It's a fantastic immune activator. Again, it's excellent for vaccines, but it's really bad for everything else. This is what we published and demonstrated very clearly a couple of years ago, and several follow-up studies showed the exact same data. Again, really good for vaccines, but ideally we would like to generate lipid nanoparticles that are not inflammatory or hopefully less inflammatory for every other applications, because you don't want to, if you want to use mRNA for genome editing or protein replacement, you don't want to induce inflammation. Another hurdle is the very precise delivery. So organ, tissue, and cell type specific targeting. It's still a big issue. If this very nice figure just shows the journey of lipid nanoparticles through the human body after systemic delivery. So after intravenous injection, at least a fraction of the lipid nanoparticles enters the bloodstream, and you can see that in the vessels. Even to the lipid nanoparticles, a bunch of serum proteins bind, and this is called protein corona. So there is a protein corona formation, and this will profoundly determine the fate of these particles. There are at least 3 different mechanisms that clear lipid nanoparticles. So macrophages can clear them, and obviously there is elimination by the liver, and also there is elimination by the kidneys. But at least a fraction of the lipid nanoparticles will leave the blood vessels and reach the target cells. The target cells endocytose mRNA LNPs, and then they, after endosomal escape from the endolysosomes in the target cells, a fraction of the lipid nanoparticle and also the mRNA, but primarily the messenger RNA, enters the cytosol of the cell where protein production occurs. But again, this is only a tiny fraction of the full dose that is given into animals or humans. So the biggest issue is that most lipid nanoparticles are apolipoprotein E, ApoE dependent. Apolipoprotein is a low density lipoprotein, and the problem is that we have tons of it in the bloodstream. So most LNPs can bind ApoE, and then the ApoE is primarily taken up by hepatocytes in the liver that are equipped with the ApoE receptors. And the problem is how can we avoid the liver? How can we avoid liver delivery? So people developed a bunch of different methods. So somehow we need to figure out how to target it. I'm just showing you 3 different versions. The first one is called passive targeting. So you can make a particle smaller or bigger if you want to make sure that it stays in the blood vessels for a longer time, or in the vessels of an organ such as the liver. You can make them. You can control the size. The other type of targeting is called active targeting. So you can add cell type specific antibody to the surface of the lipid nanoparticle, and then you can target this specific cell type in different organs, such as in the kidney or in the lungs. And the third version is that you can play around with the charge of the surface of the surface of the lipid nanoparticle. So different serum proteins bind to it, and different serum proteins can also have some kind of a targeting function, and they can target the lipid nanoparticles into different organs and different cell types. So this is just a couple of examples that I wanted to show you. So again, I really wanted to tell you about not only the successes, but also the most important problems, the most important obstacles that we need to overcome. But again, the future is, in my opinion, it's very bright. So let me finish up my talk with just 2 more slides. So this shows the various applications. Again, you can see that The major area is probably infectious disease vaccines. There are dozens of clinical trials against infectious disease infectious disease vaccines, but people started to use it for to target metabolic and genetic diseases, also cardiovascular and other types of diseases as well. So again, we have almost endless possibilities. I don't believe that mRNA will solve all the medical issues. I'm optimistic, but I'm not that optimistic. But I think it will be very useful for many diseases. And again, there are many companies that are formed. This is just a tiny fraction that shows this is the RNA lane here, and these are only the companies that have already clinical products or clinical work, and there are many in phase 2 and phase 3. So this makes me pretty optimistic, and most probably there will be more products that will be approved for human use in the near future. So with this, I'm going to stop here, and again, thank you for coming and listening. If you have any questions, or if you believe that I can be useful for you in the future, then feel free to reach out to me. And again, thanks for your attention, and I'm happy to take any questions, either in English or in Hungarian. It's up to you. Thank you.
Well, thank you very much for this truly inspiring lecture. I must say that I'm from the chemistry side, but I really did enjoy your talk, and I could follow all of it. So that's a compliment, I think. Maybe if I may, let me ask my questions first, okay? And after we can go to the audience, because you mentioned the LDL receptor as a possible problem in targeting, but is that really that big of a problem, or can that still be good in certain cases? Because I know of some nanoparticle platforms that specifically target LDL receptor based on the notion that LDL receptor is upregulated in certain types of tumors. Many tumors, they have a very high LDL intake, and maybe the liver is not that big of a problem in those cases, or is that the case?
I think,
what do you think about this?
I think it can be useful in some cases, but the problem is that so from that point the delivery will not be specific, right? So the problem is that the liver still it will be a sink, basically. It will be a waste because maybe 80%, 70% of your injected particles will be targeted to the liver, and maybe, let's say, just 3% will go to the tumor cells. So you want to be as specific as possible. And again, we have a bunch of projects in which we target the liver to treat fatty liver disease and stuff like that. But again, so for those projects, liver targeting is just fantastic. But for everything else, it's a problem.
Maybe one more question about the targeting still. What about post-translational modifications and their cell type specificity, or maybe tissue specificity? So maybe by selecting an appropriate protein, you will already have some specificity of it ending up or being produced in the cells or tissues that you want to, or
so that's one of the reasons why this active targeting that I was showing to you is useful, because you can attach an antibody that is specific to a cell type, a cell surface protein, and then you can target the particles into those specific cell types. The problem with translational modification, it's not exactly clear for me how you could use it, because you need to target the mRNA, and then the host will make the protein, and that protein will be post-translationally modified.
If the protein is made, let's say, in a lot of places, but doesn't get the appropriate post-translational modifications that it should get to function, then in a lot of places, okay, maybe it will become as a sink, but the effect might still gain specificity from the functional protein ending up in the places where you want it to.
Yes, it's possible, but I think it happens relatively rarely, particularly if only a tiny little fraction will go to the place we want it to go.
Okay.
Thank you so much.
Thank you very much. I have actually a lot more questions. It's
we
really
can,
very
we can
inspiring.
talk about them.
However, let me give the opportunity to the audience. Maybe if Monica will have to.
Sure. Thank you very much, Moni.
I'm really, really happy that you came and you have joined the Academy as a international mentor. So
thank you so much, Moni.
We will gain a lot of knowledge and a lot of possibilities with having Professor Pardi in the Academy.
Thank you so much, Moni. See you.
Okay, so anybody from the audience with questions, comments, please.
Yes.
Go on. Maybe you will need to use the microphone so that online others can also hear what you're asking.
So my name is Márton Cikály. I'm a third-year PhD student here in the Biological Research Center. And I'm really interested about how your lab works. So you mentioned that you really enjoyed this very small lab that you could really learn from the mentor. What about your lab? Is it the same? So you also wanted to actively have a small group that you enjoyed?
Thank you very much. It's a great question. Or a very important point. Pretty much, yes. So my lab at the moment, it's pretty well funded. Hopefully it will not change with time. And I really wanted to hire very motivated, talented people. You don't have to be a genius. It's enough. I'm not a genius either. But I always worked very, very hard, and I was always very motivated, and I think that's much more important. But I usually say that it's much better having smart and very motivated, hardworking people rather than lazy geniuses. So that's the strategy that I'm following. My lab, just to give you a straight answer, I have 8 people in the lab as of now. I could hire more, but I don't want to, because that's the number of people that I can really mentor, and I want to be a mentor. So I don't want to just have people in the lab doing random things.
100 people,
exactly, exactly. And I just don't see how I could manage 15 people or 20 people. So that's the reasons. And my lab really functions as a team. So my people like each other, they go out for dinner without me, and they talk about me behind my back, which is great. It's a good sign. This means that they like each other. I don't say that they are, best friends, but they work together, and it's totally natural that they help each other. There are no fights within the lab, and I think this is the biggest strength of my laboratory. And everybody is very hardworking, so that's how I think ideal lab, at least for me, functions, and again, we publish more than many people, many other labs that are much bigger, because we are very focused, we are hardworking, and we try to spend the taxpayers' money in the best possible way, and this works pretty well for me.
Thank you. Now I would have another question. Do you also work with pathogens yourself in the lab, or you just develop the vaccines?
What do you mean?
So is your lab have access to biosafety level, I don't know, 2-3 labs, and do you do the pathogen work as well?
So if you develop vaccines, you need to work, you need to see if a vaccine is protective. So we have BSL-2 capabilities, which is enough for most influenza, not all influenza viruses, but most of them, and we have a big influenza virus vaccine program. So the short answer is yes. We can go up to BSL-2, and everything else that is above BSL-2, BSL-3 and 4, we have collaborators. So in this case, we just send the vaccines to the collaborator, and they have animals in A, BSL-3, 4. They immunize the animals, and then they can challenge them and see if the vaccines are protective or not.
Thank you very much.
So that's another thing that I just want to mention, that my lab is very, very collaborative. So we have more than 50 collaborators in 30 different countries. Many of them, I'm very proud of it, many of them in Hungary. Absolutely amazing projects, very, very nice collaborators. And this is great because we have access to pretty much anything that we want.
Thank you very much.
Sure.
Further questions?
Hi, my name is Ákos Kovács. I'm a second-year PhD student in the program. and I think it's quite natural that eukaryotic organism makes an immune response to any kind of nucleic acid delivery device, but in most organisms there are some kind of extracellular vesicles and exosomes that can deliver smaller RNA types. Would it be possible to design lipid nanoparticles according to these extracellular vesicles that are more similar to natural inner particles?
Yes, very, very good point. People are working on it very hard, and people also would like to deliver messenger RNA by using actual extracellular vesicles. So they don't use LNPs, they use extracellular vesicles. I'm really not an expert in the field, but as far as I know, there are several very critical limitations. One of them is that extracellular vesicles are not homogeneous. So there are many, many different types, smaller ones, bigger ones, many different types. So the problem is that if you want to deliver an actual product that will be used in humans at one point, it has to be uniform. It has to be, all the experiments should be repeatable 2 million times. And that's my biggest concern with extracellular vesicles, that they are very, very heterogeneous. If you isolate them from cell, again, you will have a very, very heterogeneous population, and I'm not 100% sure if these are easily producible under, in the laboratory, for instance, as a synthetic product. Again, people are working on it very hard for the exact same reasons that you mentioned, that they are natural, but I think there are major issues that people could not overcome as of now. But again, the idea is great, and this is a very active area of research.
Sorry, I want to connect to that question because you mentioned that these particles are hard to replicate because of the heterogeneity. But isn't medicine going that way, that to make therapies personally and not, well, so not medicines for everyone, but for a particular person each? And therefore we won't be able to replicate everything exactly the same.
Yes, but I completely agree, and this is a great point. But at least initially, you want to show that this is something that is reproducible. Of course, after that, once you develop, let's say, a special cancer vaccine, it's called personalized cancer vaccine, and the leader is the BioNTech in this field. But firstly, they need to demonstrate that the technology works. And in this case, with the extracellular vesicles, the biggest issue that, again, as far as I know, but correct me if I'm wrong, it's just not easy to make them in GMP under conditions that are necessary to make it to human medicine, I should say. This is the biggest issue at the moment. But I agree, for many applications, it really depends. A prophylactic vaccine doesn't have to be in person, like personalized, right? So it can be the same thing can be used for anybody. But certain types needs to be personalized. But again, the big issue with this that first you need proof of concept that it works.
Any further questions? Yeah.
So you have mentioned personalized medicine, and I would like to ask that if we started focusing more on that today, how much progress could we make in 5 to 10 years? So would that actually be financially viable in terms of, let's say, for a cancer treatment to just get a personalized package for someone?
BioNTech is very actively pushing. 5 to 10 years is a long time, so I think in that time frame it's probably doable, but let's see how it works. So cancer is a very, certain cancer, not everything, cancer is very heterogeneous. So we can already cure some of the cancer types, we can make pretty good vaccines against certain cancer types, but there are some others that are not curable at this point, and there are some others that I really don't see how we could make vaccines, such as pancreatic cancer. Again, in some cases you can make it personalized, but not always. But in this, just to give you a straight answer, in 5 to 10 years, I really expect to see a huge progress in this field. But obviously, I don't know the answer because that's the future.
Thank you.
Sure.
Any other questions? Yeah.
I was a first year student last year in the program, and my question is that you've talked about cancer and other kind of pathologies here, but what about autoimmune diseases? Because in some cases that's where protein structure is really essential.
So people started to use mRNA therapy also to target autoimmune diseases, but this is still in infancy. So I know about 2 or 3 publications, but those are all preclinical work, so the mouse studies. So I think there is still a lot to do, but again, people are thinking of it and start to use it. You know, we'll see how it works, because many times something works really well in animals, and then when you use it in humans, it's just not as good as it is in animals. But again, it's an active area of research.
More questions? Yes.
So on one of your slides, you've presented the modifications of the RNA, and then you've said that you've used one certain type of modification in the COVID vaccine. I think that was the pseudo.
Exactly.
Yep. And I might not have caught it, but why actually did you use that particular one if there were other modifications that produced lower inflammation?
It's a very, very good point. So you need to keep 2 things in mind. One of them is the level of inflammation used by, let's say, modified RNA, and the second thing is the level of translation. So there are some modifications that make the RNA pretty much non-inflammatory, but the problem is that some of those modifications make the RNA also non-translatable. So you don't produce protein. You need to find a middle ground. You need something that makes the RNA more highly translatable and pretty non-immunogenic. And pseudouridine is just a good compromise.
Any other questions? Then maybe I ask one more.
Okay.
Concerning therapeutics, what is your vision about the future? Where could we possibly have the breakthrough? What do you think? What is the most promising? There's so many, I know.
I think there is one application that uses VEGF-A to treat patients with heart attack. And that is in phase 3. And as far as I know, the phase 3, some phase 3 data was already released, and it looks pretty good. I think that's the most promising at the moment, but I don't know if it's going to be a drug or not. But there are many different.
How about insulin, for
I'm
example?
sorry?
How about insulin?
Why would you use? I wouldn't use it. I wouldn't use RNA.
No?
It's much cheaper. So insulin, it's a protein. It's a very short one. It's very easy to make it. We already have a fantastic system, and you can make it for pennies. So I wouldn't use RNA. It's very important. Again, I love RNA, and I really like RNA therapy in general. But again, I don't want to be too biased. I'm biased, but I don't want to be too biased. And if it doesn't make sense to use RNA, then just let's not use it. You also need to keep in mind there are practical considerations, and the product should be as cheap as possible. And I don't think that you can beat insulin as a recombinant protein if you use RNA. It's just good enough.
It's good enough. Yeah. One more question there.
So for insulin, it doesn't make that much sense to use mRNA, but perhaps for other, are there other candidates currently that we cannot easily manufacture otherwise?
Oh yeah, there are many. So that can be one very important advantage of using RNA, particularly for proteins that have a very short half-life. because there is continuous protein production from the RNA. So that's one of the clear advantages if you want to use mRNA for protein replacement. There are many candidates. If you check out the website of Moderna, for instance, or Pfizer, or BioNTech, or CureVac, you will see many different candidates, even in clinical phases.
Thank you.
Sure.
Any more questions in the audience? Yeah, back there, please. Just hand over the microphone.
But if you want to replace something that's missing, like insulin with RNA, RNA gets degraded over time.
Yes.
So how often do you have to re-inject yourself with RNA for this to work?
I don't know. It really depends on the RNA that you are using, because different RNAs have different half-lives. And it also depends on the application. It also depends on the dose that you are using. I cannot really answer this question, but what I can tell you that if you use a relatively low dose, let's say a couple of microgram, 3 to 5 microgram in mice, then the RNA is there for 10 days. Again, it really depends on the mRNA. It really depends on the nanoparticle. It really depends on the route of delivery. So there are many different variables, and the combination of days will determine most probably the frequency of injections.
Yeah, and if it's about injection, do you necessarily have to inject yourself, or are there attempts to make a Peros delivery system? Because somehow
people are trying to develop. Yes, people are trying to develop patches that can be used, but it's not that easy, and there are several limitations. People also want to use it for intranasal delivery. Again, I don't have the time. It's a long thing, but
it's a pulmonary lot of.
Yes. Again, there are problems with that, but it's ongoing work.
I see. There's one more question over there.
Are there any attempts to target bacteria with mRNA vaccines, or?
Yes, bacteria, and we just started a fantastic collaboration with Professor Attila Gácser, and we target fungal diseases, because there are basically no vaccines against any fungi. So the answer is yes. Obviously, the major focus is viral diseases, but people try to target bacteria, fungi, and even parasites.
And they are all vaccine-like attempts, or are there any like antimicrobial peptide production?
Very good question. Yes, we also try to develop antimicrobial peptides, but it's also a very complicated problem, because these are very short, so you need to worry about many different things. You need to apply it specifically. You need to figure out, because you probably know that these antimicrobial peptides are made as a peptide, so they need to be cleaved several times. So it's not easy.
Sometimes non-ribosomal peptides.
Exactly. You need to figure out how to make it secretable. So, but yes, people are working on it. I also have a couple of collaborations on this.
Great. I would be happy to study how the evolutionary dynamics change when you apply as an mRNA antimicrobial peptide, how it changes versus the classical Peros delivery of antibiotics. It's really interesting. Thank you.
Sure.
Further questions? Yeah, there's one more.
So you've mentioned the financial threats the current administration poses, but I'd be interested in another aspect of it, like that some politicians actively erode public trust in your field, in your work, like making anti-vaxxer sentiment mainstream. Do you have any plan or any strategy to cope with that? Like, do you plan to put more emphasis on popularizing science?
Well, I think it's always important to explain people, particularly people who have concerns about any therapeutics or any medicine. I should say it's not only messenger RNA, it's everything. So we would need to invest some energy, but to be completely honest, I think if you are a busy scientist, you just don't have the time to talk about all of these things. And I'm trying to focus primarily on those people I'm focusing on anything. I'm focusing on my work, because I really don't think this is strictly my job. Of course, if somebody reaches out to me and wants to learn more about mRNA vaccines, it happened many times that somebody just sent me an email, a nice email, and he or she wanted to learn about mRNA vaccines, and I always answer. I always try to provide some guidance and also some literature. This is what I can do, but I simply don't have the time to deal with those people, and again, I clearly don't think that this is my job to convince people about this. But again, it's very, very important, I agree. But I can do to make sure that my lab is still well funded, that I started to diversify my funding sources. So I apply to many grants in the European Union and elsewhere where basically mRNA research is not punished.
Thank you very much.
What is your personal favorite among the many projects that you are
I have many.
Working on?
They are
many favorites.
all my children. So you don't have a favorite child.
Oh yeah, that's a great point. Any further questions, maybe? Yeah.
I'm Gergő Bíró, and thank you for your inspiring presentation. We talked about cancers and autoimmune disease, and what about other non-communicable disease, for example, cardiovascular disease or neurodegenerative disease? What do you think? Is the mRNA technique applicable in these cases?
I think so. Again, most of these there are exceptions. So the VEGF-A therapy that I just told you, it's in phase 3 clinical trials, but many others are in preclinical or clinical phase. I'm pretty optimistic that we will be able to use it at least to treat certain diseases. I think people now started to use RNA to target pretty much any diseases that you can imagine. And again, I don't think that it will work for everything, but hopefully it will work for many.
Further questions? If there are no more, then thank you again.
Thank you so much.
Amazing talk, and thanks to all.